Good afternoon, everybody. Thanks for joining. My name is Ahmad Bukhari. I am faculty at St. John's University. Can you hear me?
Yes.
Okay, thank you. Well, thanks for joining. So I'm very excited to introduce our speaker, Dr. Humayun Sharif. Dr. Sharif did his PhD from Max Planck Institute of Biochemistry, Germany. And then his postdoc at Harvard Medical School, where he's working on advancing structural biology and biochemical techniques to study host pathogen interactions and innate immune responses. Thank you so much, Dr. Humayun Sharif, for joining and for your time. Over to you.
All right. Thank you, Sayyid Ahmad Bukhari, and thanks everyone for joining. So my title of the talk, as it says, seeing is believing. This is what we do as a structural biologist. We try to solve the protein structures and try to see the atomic level model of the proteins and try to see how it does its function. And we try to make rational hypotheses and try to see if we can make better drugs if these are involved in special kind of diseases or some some immune for example some immune diseases. So I'm currently at Harvard Medical School working as an instructor, and my work, as Ahmad just described, is mostly involved in innate immune system and immune defense. So I will take you through the introduction and introduction of the immune system, and then I'll take you through the innate immune system, and then I'll show you how we solve the structure and three-dimensional protein structure, and then I'll take you through our results. So a balanced immune system is required for a healthy lifestyle. Any upregulation of balanced an immune system can lead to autoimmune diseases, for example, or sometimes, as you can see on your skin, sometimes you have this allergic reaction, and that's because of the external threats and because of the upregulation of your immune system. But sometimes the downregulation of the immune system, just like in cancer or during the infection, can also lead to a lot of disease. So what you need is a balanced immune system where it's not upregulated, it's not downregulated, it's at the perfect balance. So of course, if the balance is disrupted or any dysregulations, especially in the innate immune system, can lead to a lot of diseases. So innate immune system is the first line of defense in the human body, where it can check for external or internal threats. Whenever it finds any threat, it will react and do its function, and its function is basically to stop the infection. So the dysregulation in innate immune system, that is basically upregulation or downregulation, can lead to diseases such as inflammatory bowel disease, joints disease such as arthritis, metabolism disease such as diabetes. So these are the sterile inflammatory diseases that occur because of the activation of these overactivation of these immune system. But also, if there are some mutations, that is a change in our genetic makeup, any change in the genetic makeup can also lead to diseases. These are called autoinflammatory diseases, and some of them I will take you through in my slides that are following up. Now, I was talking about the innate immune system. So these innate immune system, they have the specialized cells. These cells are equipped by pattern recognition receptors. These pattern recognition receptors, they are looking for patterns, some external patterns that are called PAMs, pathogen-associated molecular patterns, and these arise as a consequence of a pathogen attacking our bodies. For example, a pathogen, bacteria or virus, when they attack our body, they have specialized fragments or something on their surface, just like you can imagine coronavirus. They also have specialized the surface, and there are specialized proteins, and these proteins are recognized by our innate immune cells. Now, if it's not external threat, if it's an internal threat, that is called damage associated molecular patterns. For example, in the cancer cells, the necrosis, or if the cells are dying, or some other factors that are arising from within our body. So this can also activate these innate immune response. So what happens is that I told you there are specialized pattern recognition receptors. They're either extracellularly or are present on the surface of the cells, or they are internal in the cytoplasm of the cell. The cells have this specialized boundary called the cell membrane. So if the threat is present outside the TLRs, the toll-like receptors, they can look for these threats and they can internalize, and then they can start the cell death pathway or the innate immune response. But today's talk will be focused on the NOD-like receptors. These are internal receptors that are present inside the cells, and they're always looking for danger signals, either PAMs and DAMs. So what happens is that once the NLRs, or NOD-like receptors, they identify external or internal threats, they start making these activation of the caspase-1, which is a protein that can activate the cytokines. So cytokines are, the alarmins. So basically, the alarmins are like, they will trigger immune response. So what happens is, so when our innate cells, they identify external threats or internal threat, they will mature these cytokines or the alarmins, and these alarmins then will tell other neighboring cells that there is a infection site and you should get rid of these cells or these microbes or pathogens, for example. So our, what we in the lab, in my current lab, we try to understand the inflammasome complex, and we try to see how these NOD-like receptors, they arrange in the large molecular assembly, and how they look in the three-dimensional view. So if you can understand, there are three types of proteins that are required to make an inflammasome. So one, the sensor proteins are mostly the NOD-like receptors, the adapter proteins, and the effector proteins. So the sensor proteins, when they identify a PAMs or DAMs or any threat, they will start making long oligomers, or multiple copies of these sensor proteins, and then they will activate the caspase-1, which is an effector protein. So once the effector protein gets active, it will active other proteins. So as I told you, IL-1 beta and IL-18 are the cytokines. These are the alarmins, which will alarm other cells or the neighboring cell that this is a site of infection. So what it does is, it will cleave, so caspase-1 will cleave the Gasdermin D, it will go in the cell membrane of the cell. It will rupture the cell membrane, open up the cell membrane, so that these alarmins can be released into the periphery of the cells. So either cells can survive during this whole process, or sometimes cells, due to the leakage of these alarmins, and also because of the flow of the the fluids from back and forth, what happens is that the cells swell and they just die. And the process is called the pyroptosis. So it's a programmed cell death pathway in which the cells are dying on its own in order to survive, in order to save other cells. Basically, when the cell dies itself, it will release these alarmins and will tell other cells, neighboring cells, that this is the site of infection. I have killed myself, but you can get rid of it. So other proteins then come and they try to save or try to kill this virus or bacteria, for example. So my first part of the talk will be focused on the NRP3 inflammasome, and I'll try to tell you how another protein, NEX7, helps it in activation of these inflammasome. So as I told you, these NLRs, they get activated, or the inflammasome get activated by a lot of DAMP, which is the internal signals for death, or the disruptions in the cells, or the PAMs, that is the patterns from the pathogen. So there are almost never-ending list of PAMs and DAMs that can activate NRP3 inflammasome. So our focus was in order to understand which of these PAMs and DAMs they bind directly to NRP3 so that it can make inflammasome. So we started off with purification. So what we do as a structural biologist is that we purify the proteins that we are interested in, and we try to make a complex with other proteins that these proteins interact with. So sometimes these proteins, when they interact with the protein, other proteins, they make the complex, and that is required for downstream processing. This is what happens exactly in NRP3. While we were working on NRP3, this is the domain architecture of NRP3, and I will show you how it looks like in 3-dimensional. So you can just imagine this is a polypeptide of the amino acids, where these amino acids are divided into subdomains. So these domains are individual folded protein complex. It's a folded protein 3-dimensional assembly where different domains make up to make a full-length protein. So we made a complex of NRP3 plus NEX7. It was described in 2015 that they make complex, and NEX7 activates NRP3 inflammasome. So we purified these proteins. This is just the purification of this is how we purify, and we see on a size exclusion chromatography in which we put the protein, crude sample of the protein, and the system basically extracts on the base of the mass of the molecule. So earlier elution, or if the protein comes out early, that means the protein is larger. Smaller protein comes out later. So if you make a complex, that means it's a bigger protein complex. That's why it comes out a little earlier as compared to NRP3 alone. So once we purify these proteins, what we see is we just make the grids, small grids, and we see under electron microscope how these proteins are looking. So you can see these individual small dots here are individual protein molecules. So our goal is to basically collect as much data so that we have enough these small particles so that we can make a 3-dimensional model. So I'll take you through how we do it. So this is a cryo-electron micrograph, and these are small particles here. So in order to get a 3-dimensional view here that you're seeing at 3.8 angstrom, what we did was we collected these micrographs, almost 15,000 micrographs like these, and we then extract these small molecules or the small particles from these micrographs, and then we stack them together and make the 2D class averages. So this is the 2D class averages. So this is the averaged molecule where you can see the NRP3 with the NEX7, these individual 2D class average, shows there are different molecules in this image that sit like this, and that's how we made a composite of it. So in this way, you can imagine, because our proteins are 3-dimensional, right? So you can so these proteins can sit on the grids in different orientation. This is what exactly we see in 2D class averages, where the protein is fitting on a different orientation, and that's how you just you have these 2D class averages. So once you have these 2D class averages, almost 160,000 particles, we make a three-dimensional classification. So since these 2D class averages are from different views, idea is that we can collect 3-dimensional or 360 degrees data on all these molecules, so that we can merge all these 2D class averages to make a 3-dimensional model. So the idea was the same thing, and we made almost 6 classes, and then we refine it through through the algorithms where we can refine the structure, and the better looking or better visualized structure is finally we take it. So class 2, for example, which contain almost 67% particle that contain almost 100,000 particles, and these are then further refined so that we can get a 3-dimensional view or 3-dimensional model of the density. So basically, this 3.8 angstrom resolution means that it is pretty much good that you can start seeing individual atoms in 3-dimensional sphere, and where you can start building the protein model. So this is the whole density here. So in 3-dimension, this is how the protein complex looks like. As I told you, NRP3 has these 4 subdomains. These are colored here also in the same way. So you can see they are folded together and makes like a a gel. They are gelled together, and they make like a globular structure called the Nash domain. And then after that, there's a loose enriched repeat, which is like in cyan here in this blue color, which makes like a curved shape. It's also like horseshoe, basically horseshoe magnet. So basically it's just like that. And together these two domains, the NASH domain, all these 4 domains plus this LRR, which is cyan in color, they can capture a small molecule, which is NEX7. It's a NEX7 protein. Here you can see in yellow the NEX7 protein. So what is happening here? There are 2 important things. ADP, it's a small molecule, which we know is an inhibitor. So basically, when you add the inhibitor ADP in the cells, it will lock the protein and it will not allow it to activate. And then it is also bound with the NEX7, which was supposed to be its activator. So we made a hypothetical intermediate or hypothetical active state. This is the structure that we have solved. But by using homology model, we know that there is another protein that was solved in our lab, and this was solved in an active state. What we understand from this is that 2 domains, NPD and HD1, green and blue domain, they need to rotate 90 degrees so that they open up. So once the NLR structure opens up, that means it's an active state. So active state then can oligomerize, basically it can bind to other molecules and make this ring-like structure. And this ring-like structure is an active state. So we made a hypothetical active model, and we were able to see, okay, these are the interaction surface that we do not see on NRC4. So we wanted to see how it looks like in cells. So we changed these residues, these interaction surfaces, and we change the residue and we see the effect of these changing residue in the cell. So we make the mutations and we see that these mutations can no longer activate the inflammasome in the bone marrow derived macrophages, which is the innate immune cells in the mouse. So what we concluded from this is that NEX7 is required for activation of NRP3 for oligomerization. And that's why that's what was discovered in 2015, that NEX7 is required for activation, but its molecular details were not known. So we were able to give the very first time how NEX7 will mediate the activation of NRP3. And that's how, we were able to present our results in 2019. And then, as I told you, the mutations in these proteins can lead to a lot of diseases, such as cryopyrin-associated periodic syndrome, CAPS syndromes, are basically the syndromes in the infants and adults. What happens in these patients is that they have rash from very early childhood. They have central nervous system inflammation, and they also have the periodic fevers and joint pains. So we wanted to understand how these changes in the genetic makeup lead to activation of these inflammasome and basically then lead to these diseases. So these are the mutations that have been already reported from the patient's data. And what we were able to do is we were able to model these mutations onto the surface of NRP3, the structure that we had solved. And here you can see that these mutations either abolish the inter-domain interaction or they can abolish the ADP molecule. As I told you, ADP molecule is required for keeping the protein in closed conformation or in an inactive form. But you can imagine if there are mutations in these activation pocket, the protein cannot remain in an inactive form, but it will always be active. So this always active means there will be alarmins that are released into the periphery. There will be alarmins always present in the body. That means body think there is a threat, and then it can lead to periodic fever, for example. But in general, what is really happening is that there is a mutation in the protein that keeps the protein in an active form, and that's why you see the inflammation in these patients always present. So in the end for the first part of the talk, what I had shown you is that we were able to solve a 3-dimensional structure of NRP3 bound with the NEX7. What we hypothesize is that it will require activation by some unknown factor or by ATP exchange. Once it gets activated, NRP3 oligomer is formed, and this oligomer can lead to activation of inflammasome or can activate the release of alarmins or can also lead to programmed cell death. In the next part of the talk, I will actually just talk about NRP1, and I will show you how another protein, which is a DPP9, can act as a regulator of inflammasome assembly. Now, these inflammasomes are different, and sometimes the activation mechanism is different. So, and sometimes the domain architecture is also different. So, especially for NRP1, so I will be focused on NRP1. You can just ignore this cardate for a while. So NRP1 has the PYD, NASH, and LRR as the domain architecture is very similar to the one I showed you for NRP3. It has these NASH and LRR domain. But amongst all the 22 copies of NLRs, or 22 NLRs in the human protein or in a human body, NRP1 is the most unique because it has fin domain on the C terminus and then the CAR domain. These two domains are very unique, and they go on. So the fin domain go on to have autoproteolysis. Basically, it will cleave itself. It will cleave itself, and the two subdomains, ZU5 and UPA, are kept together by non-covalent interaction. So basically, they have the traction with each other and they are kept together. So this is a very important point, and I will take you through why it is most important. Now, contrary to NRP3, in NRP3, in the model that I just showed you, the oligomer model, I showed you that NASH and LRR are required for making this ring-like structure. But for NRP1, it is different because NRP1 active structure only requires the UPA and the CAR domain, the C terminus. It does not require the N terminus where the NASH and LRR are required for activation of inflammasome. And that's why it's the most unique. And understanding the mechanism or architecture or the molecular details was very important. So, and why it is important, because there are some mutations in the protein, the genetic mutation that can lead to skin inflammation, or cancer susceptibility syndromes, especially a mutation in the patients containing the P1214R. This can lead to arthritis, this kind of joints inflammation, and disc keratosis, some skin deformations. So we really wanted to understand what are the molecular details of these mutations and how we can tackle if we are trying to understand the mechanism, and if we are trying to make better drugs against these for these diseases. So we went on to study NRP1 inflammasome. So NRP1 inflammasome, or domain architecture I showed you earlier, and then the DPP9 is the regulator. So DPP9 is a dipeptidyl peptidase. Basically, it will cleave the peptides in the cell. So whatever the substrates are. So coming back to NRP1, as I told you, the fin domain goes on to have the autoproteolysis. So it will cleave itself. We don't know the exact the rationale for it to cleave itself, but the cleavage has its own benefits. This is what I will show you. So in the resting stage, when the cells are not doing anything, when they're there's no infection, the DPP9 will bind to NRP1 and keep it in a closed conformation. So these domain architecture I've shown you in small spheres is showing representing these individual domains. So we know from literature that the DPP9 binds on the fin domain. ZU5 and UPA domains are here. So if you can disrupt this interaction of NRP1 with the DPP9 by the presence of PAMs and DAMs, if for example, if the virus or bacteria attacks the cells, it will disrupt this interaction. Just remember, DPP9 is a regulator. Basically, it keeps the NRP1 in check. But if this interaction is disrupted, that means NRP1 inflammasome can get activated. So it's like a host defense mechanism. So PAM or pathogen-derived pathogens, when they are present in the cytoplasm, this interaction will be lost. And that's for as a host strategy to counter these pathogens, it will activate NRP1 inflammasome and will release this in organism cells. So how it does is that, and we can also disrupt this interaction by small molecule inhibitor like a drug molecules. What happens is that if we disrupt this interaction, NRP1 molecules or NRP1 proteins are targeted to proteosomal degradation. So proteosomal degradation is a proteasome is a protein which degrades other proteins. So by disrupting this interaction, this proteosomal degradation gets activated, proteasome, which is like a small, like a, a big protein, but it will capture the N terminus of the protein, just like the PYD, and it starts chopping off or degrading the protein. So basically, it starts eating the protein from here on the N terminus. But when it reaches the autoproteolysis site, which is what I told you, it's very important because these Zoo5 and UPA domain are kept together by non-covalent interactions. Proteasome, for the proteasome degradation, it requires a continuous polypeptide chain. So, but whenever there is a break, proteasomal degradation or proteasomal activity will stop, and it will release the C-terminal fragment, UPA and the CAR domain. These C-terminal fragments, they have these CAR domains, which has the intrinsic ability to interact with the other CAR domain to make long filaments in the cytoplasm. And that is required for inflammasome activation. This is what I just told you, that these proteins, they oligomerize and then make these long filaments that activate these alarmins. So we went on to study these interactions, NRP1 with the DPP9, and we went on to have the CRI-AM structure once again for NRP1. So this is the gel. Basically, it shows you the quality of the protein. The cleaner the protein, the better it is for CRI-AM studies. So here you can see individual protein. This is what we know already from our expression system. We take the protein sequence, we recombinantly express these protein in the mammalian cells, just like human embryonic HEC293 cells. So basically, what we are using the human cells for growing artificial protein. So once we make artificial protein in a larger quantity, we purify the protein from other, background protein, and and then we try to see them under the negative stain or under the microscope. Exactly what we did for NRP3, we also did for this NRP1 DPP9 complex. We made CRI-AM grids, and then we collected data, almost 8,000 micrographs, just like that. And then we clustered these individual particles and make the 2D class averages. So you can imagine these 2D class averages of almost 800,000 particles. And then we were able to see in different views how the complex looks like. You see, it's like like a butterfly kind of structure. And in these 2D class averages, you can already see that there are some finer details. So we then also collected some more data, because the problem was we were lacking. So if you can see this sphere on the left-hand side, the individual blue dot means the view from that angle. You can imagine in the whole sphere, there are a lot of missing angles if you see in the bottom of the sphere. So if you have missing angles, then you do not have enough information to make a three-dimensional model. So what we did to circumvent this problem, we tilted our sample in the microscope, and then we collected more data. So the tilt helps in getting more angles, of different angles, and that sometimes helped in getting more angles and eventually a better data set. So we were able to get almost all sphere. Here you can see now the whole sphere is filled up with all the images from all different angles, and we were able to get a 3.6 angstrom resolution, which shows a pretty good quality of the data that can be used to build the model. So once we had this three-dimensional model, just like that, the three-dimensional density, electron density, then we fit in the individual amino acids, and then we look for three-dimensional view. And this is a three-dimensional view of those 2D class averages. So you can see already from here, the 2D class average in the previous slide looks like, this just like the central class, you see, just like that. Our model, if you see in the three dimension, looks just like that 2D class average. So once we were able to get this density, this is the density, CRI-AM density, and this is a structure, atomic structure fitted in the density. So what the idea is that we try to fit in the density or the individual amino acid in the density, and by keeping the stereochemistry and all the bonds in check. So in this structure, what we are seeing here is that the green is the dipeptidyl DPP9 protein, and so these are two protein molecules. So here you can see it's a dimer. So there are two protein of DPP9, And each monomer of the DPP9 binds to 2 copies of NRP1 called the site 1 and site 2. Site 1 contains the full-length protein, full-length fin domain containing Zoo5 and UPA. Here is the color coded as in this cartoon. And then the site 2 contains only the UPA. So first thing that struck us was why the site 2 cannot accommodate the Zoo5. So as you see, the site 1 has the full-length fin domain containing Zoo5 and UPA, but the site 2 has only the UPA. So our first question was why it cannot accommodate Zoo5. So then we did a molecular modeling approach in which we modeled the Zoo5 onto the site 2. And so here is the site 1, and this is the site 2. This is a copy we only see, the UPA 2. But if we model the Zoo5, we can see that there will be clash with the Zoo5 and also with the DPP9. That means site 2 cannot accommodate Zoo5. And that also means probably this is the outcome of the proteosomal degradation in which the C-terminal fragment containing the UPA and the CAR domain is liberated. This is the proteosomal degradation fragment. You can see that here on the site 2, we can see the UPA in the present, UPA present on this site 2. So what we have concluded is that since, as I told you in the activation mechanism or activation model, if these are kept alone in the cytoplasm, this card and the UPA, this C-terminal fragment, these will make inflammasome, or they can oligomerize and activate the inflammasome. That is a problem because if in a resting state, if there is no threat, these C-terminal fragments should be kept or should be sequestered in order to avoid any inflammation. So in order to avoid this inflammation, and DPP9 sequesters or basically binds to this C-terminal fragment and keeps it on the site 2. So basically, it is sequestering the activation of NRP1 and keeping the protein in check. So the overall model of the site 1 and site 2 is slightly different. Site 1, as I told you, contains the full-length protein, has full-length fin domain, having the UPA1 and the Zoo5. Site 2 has only the UPA, and then there is a difference of the structure. N-terminus of this fragment would restructure itself and then insert itself into an activation tunnel of the DPP9. So the proteins have the active residues where they can do the enzymatic activity. For DPP9, the enzymatic activity is that it will cleave the protein or it will chop the protein. But here in the NRP1 example, DPP9 only function for the DPP9 is that it will sequester this C-terminal fragment to avoid any inflammation. Now, if we just zoom in onto this part where UPA extends itself, go into the activation tunnel. So we call this activation tunnel because serine 730 it's an amino acid which is an active protein residue, and this residue is required for cleaving the target protein. Here, the target protein is the NRP1, but DPP9 cannot cleave it because the only function here of DPP9 is that it is holding on to this UPA2 so that it does not get activated. So there are very important things happening here in this structure. You see that N-terminus of the UPA is kept by the DPP9 active site, and then there is a mutation. There was a mutation that I showed you in the initial slide, that proline 1214 to arginine. This is a mutation that can lead to arthritis and disc keratosis. Now, what we understand from our structure is that if the proline, instead of proline, arginine is present, as have been seen in the patients containing these mutations. If the arginine is present, DPP9 cannot sequester or cannot hold on to this mutated protein, and that's why there will be liberation of the C-terminal fragment, and that's why there is inflammation in those patients containing these P1214R mutation that leads to arthritis and disc keratosis. So our structure was able to rationalize how this activating mutation can lead to arthritis and disc keratosis. The last part of my talk, I will just take you through quickly to a very interesting aspect. So this is a cellular assay. So the red bar, the black bar is a DMSO, it's a control. The cells are not treated with anything, but the red bar shows, the release of LDH, which is the death hormone. For example, you can imagine if the cells are dying, more LDH is released. So if we treat the cells with the valborapro, which is a small molecule inhibitor of the DPP9, what we see is that there is a lot of cell death happening. So what we imagine that maybe this small molecule or this drug molecule basically inhibits the NRP1 interaction with the DPP9, and that's why there is more and more cell death, and there is more and more inflammation happening. This is exactly what happens in the cells and also body if you treat the cells with the valborapro. So we were able to solve the structure in the presence of this drug valborapro. One thing we readily see in our densities, just cardium density, is that the site 2 or UPA density is almost gone. So we were not able to model the site 2 here in this structure. So what is really happening with the valborapro, in the presence of valborapro, the site 2 cannot be accommodated. That means because valborapro or this drug molecule is present in the active tunnel residue or the activation site of DPP9. And once the one molecule is bound there, DPP9 or NRP1 N-terminus of the site 2 cannot be thread into because there's already a small molecule present. And that's why, because of the presence of this drug molecule, NRP1 cannot bind, and that's why you see a lot of cell death. This is in contrast to what we have solved, what I showed you earlier, the APO structure in which N-terminus of the UPA2 can bind in this activation tunnel. But if the cells are treated with the drug, these UPA2 cannot be bound, and that's why you see a lot of cell death, and that's why you see the release of this C-terminal fragment. So in the end, this is just the last slide of my talk. We were able to update the model in which we say that in the resting stage, DPP9 bind to NRP1 on the site 1, and once the PAMs and DAMs are present in the cytoplasm, they will release the C-terminal fragment. These C-terminal fragments can also be captured or sequestered by the DPP9 on the site 2 in order to avoid any inflammation. But if there is a disruption in the cells because of the presence of valborapro, or if you treat the cells with the small drug molecule, or because of the PAMs and DAMs, if the homeostasis in the cells is disrupted, this interaction of DPP9 with the site 2 will be disrupted, and these fragments will be released. These C-terminal fragments will be released. These C-terminal fragments will interact with themselves, make these huge filaments, and will activate the inflammasome, or basically can activate the cell death also. In the end, I would like to thank people who were actively involved in these projects. In NRP3, Lee, Wiley, and Venkat, and in NRP1, Cardet, inflammasome, Bobby, who's a graduate student who was working with me, and he recently graduated, and our collaborators at Sloan Kettering and also HMS. I'd like to thank everyone for listening, and I'll be happy to take any questions.
Well, thank you so much. A very, very informative and interesting talk. So the audience, if you have any question, please type, or you can unmute yourself and can ask.
Thank you so much for visiting us and giving this very enlightening and thorough talk. I guess one question that's just interesting me is, what are your plans for future work in this area?
Oh, so you see, for NRP3, this has a lot of potential for drug discovery for a lot of diseases, especially for Alzheimer's. So a lot of people are trying to solve the structure with the drugs that they are developing. Recently, just last week, Novartis, they have published the NASH domain. I showed you the full-length protein with the NASH and the LRR. They recently solved the structure of NASH domain with the small molecule inhibitor, means the drug that they have developed. So our plan is also trying to see the different drugs bound with the NASH domain. And then our also plan is to really get the activated structure of NRP3. So the model, the oligomer model that I showed you, it's just hypothetical model that we think is right. But eventually we need experimental proof. So we are in the lab. I'm currently working with another postdoc, and we are trying to get activated structure of NRP3. For NRP1, we are still trying to get structures of other proteins that bind to NRP1. For example, recently RNA has been shown to interact with the NRP1 and activate the NRP1 chromosome. So we are trying on that route for NRP1.
Very nice. So, how do you see, I mean, can you use these models? I'm asking like a data scientist point of view, that is there a way we can early predict uh, autoimmune disease, just like maybe rheumatoid arthritis at very early age, that in future that would the patient will develop that, or any other skin disease which you show that?
I think these models can help to understand the individual mutations in the patients, for example. I mean, not in the early detection, because uh, the only way is that we are able to develop better drugs if we have the information of the mutations in these patients. I think this is where the structural biology comes in, because you can visualize the individual amino acids that are contributing to diseases. So if you are making a drug molecule, and the mutation is present around that drug binding site, so you can try to make a better drug in which it will have a potent effect. So basically it has a downstream effect. I mean, the structural biology has a downstream effect more as compared to early detection, I would say, and more towards drug discovery, I would say.
All right, thanks. Any other question from audience? Okay, so if we don't have any question, we once again thanks to our speaker
Thanks.
from our
Thank you.
from our division and our university behalf. And for the audience, please stay tuned. We will have a semester break, and after the semester break, we will have the three people lineup from the industry. We'll mainly talk about the natural language processing and knowledge engineering for the next semester. Thank you for joining.
Thanks, Ahmed, and thanks everyone.
Sounds great. Have a good holiday weekend, everybody.
Bye.
Thank you. Bye-bye.
Thank you.
Nice to meet you. Thank you.
So.